Okay, so the procedure, I'll give you a quick visual summary of both procedures for experiment 1 and 2, and then we'll look at the specifics of those. They use these go-no-go tasks that I showed you before, but they're called one a cool task and one a hot task. The cool task was gender, and the hot task was using happy or fearful faces. The reason why it's called a cool task is there's nothing particularly alluring or emotional about whether these people are male or female. Okay, so in the cool task they would have gender. So you would say, when you see a male face, click. When you see a female, don't click. Okay, and then they could mix that up and they could say, right, on this trial you're clicking when you see the females and not clicking when you see the males. So it's called a cool task because it's not a particularly emotive, impulsive thing to be able to recognize males or females, whereas the hot task, they're seeing this as the cookie trial, the marshmallow trial. A happy face is something that we really want to react to. So a go no go for this would be something like, when you see someone that isn't smiling, then click go. Not smiling, go. Smiling, don't click. And then not smiling, go. And again, they could flip those around. When you see the smiling face, go. When you see no smiling face, don't go. Okay, so they did do that. They alternated which way, whether it was a go or a no-go, so a click or don't click. But that's called the hot task because they're saying that this is an alluring stimuli, someone smiling at you compared to not smiling at you. Okay. By the way, I've used these as an example. It's not the original article photos, so they probably wouldn't have been smiling. They probably all would have had the same facial expression in the cool task. Okay. Experiment 2 was exactly the same as this one. Okay, they used the hot task, but they did it in the fMRI scanner. Okay, so they were doing it in there, whereas for this one they were just measuring the reaction times and the mistakes, whether those who were high delayers were better and more accurate. They didn't click the false click. They didn't go when they shouldn't have gone, more than the low delayers. Okay. So, the specifics of it. The 59 participants consented to taking part in the behavioural version of the hot and cool impulse control task, and so that is the go-no-go tasks. They're completing the two versions, the cool version of the gender, and the hot version was the fearful and happy facial expressions. Okay, so go over your stuff and highlight those, the cool, the male and the female, and the hot being the fearful and the happy facial expressions. Okay, and they were presented to them using program laptops that were sent to participants' homes, so they didn't have to come into the lab to do it, but you are controlling and standardizing the procedures. They have to just follow a computer program through. It wasn't PowerPoint, but it's essentially a timed PowerPoint thing. It's really a program that allows you to run these go and no-go tasks on a laptop. Okay, so the little bit more detail on the procedure. Before the onset of each run, a screen appeared telling them which was the target. So for these next 20 faces, it's going to be male that you want to be the button press. The go is the male, the no-go is the female. But they did different trials where that was alternated. Participants were instructed to respond as quickly and as accurately as possible. So that was their standardized instructions. Everyone was told to do it as quickly and as accurately as they could. Each face appeared for half a second, and it was followed by a 1 second interstimulus interval. So between each between each piece of stimulus, you had 1 second to wait before the next one. So it's pretty swift. 160 trials were presented per run. Okay, so let's say you were doing the male being the button press, the go, with the female being the no-go. You would have 160 trials with that. In a pseudo-randomized order. That means what seemed like a random order, but actually it wasn't. It was 120 go and 40 no-go, but very, very difficult for you to be able to predict which order they would go in. The task was therefore a 2x2 factorial design. In other words, two conditions of go or no go, and then the two conditions of, I've just found my mouse there, go or no go, and then two conditions of the gender as well, male or female. So they did that 4 times. They had 4 different trials to cover each different outcome and to do enough to counterbalance the order of the male and female faces. They wanted to get enough trials to be able to draw strong conclusions. Accuracy and response latency data, that means the speed, the reaction time, and whether they accurately identified them as go or no go, were required in the 4 runs. Okay. And in experiment 2, the fMRI was thrown in, as I mentioned before. So it's the same as experiment 1, but they're just doing the hot task. Okay, and a kind of little aim for experiment, or at least a little hypothesis. It was anticipated by the researchers that the low delayers, okay, those who couldn't stop themselves when they were kids going for that cookie, would show diminished activity in the right prefrontal cortex. So in that frontal gyrus, you would have low levels of activity compared to the high group. Okay, so you'd have low levels of activity and amplified activity in the ventral striatum. Okay, so compared to the high delayers, they're going to have more activity going on in the middle of their brain compared to the frontal cortex. They're going to have lower diminished activity compared to the high delayers. So it was 27 participants from experiment 1. As we've mentioned, the participants completed the hot version of the task, and differences in the timing, the number of trials and the apparatus. So there were slight differences between experiment 1 in terms of how long they had and the number of trials and the apparatus, because they're in an fMRI scanner this time. Each phase stimulus was presented for half a second. 500 milliseconds. Sorry, not half a second. No, that is half a second. Apologies. Half a second each phase, followed by a jittered intertrial interval ranging from 2 to 14.5 seconds in duration. That just means the time between the different pictures isn't predictable. Okay, so you could be waiting for the next photo for 14 seconds sometimes. And is it going to be a happy face? Is it going to be a fearful face? Okay, and you're waiting and waiting and waiting. Or it could just be as quick as 2 seconds. Okay, but as a mean, it was 5.5 seconds they had to wait. Okay, total of 48 trials were presented per run, again in a pseudo-randomized order, but they only did 48 in experiment 2. fMRI scanners are expensive to run. You probably didn't spend as long running this as in the first experiment when they were doing it at home. In total, imaging data was required for 26 no-go trials and 70 go trials. Okay. The task was viewable by a rear projector screen, so at the back of the fMRI scanner, and that's known as a neuro screen, and a 5-button response pad. In other words, they had a response pad that they could say whether it was a go or a no-go. Okay, and that recorded the reaction times as well. So 2x2x2, which essentially means go or no go, high or low delayers, and whether it was the fearful or happy faces. And so it's a repeated measures, or which is also known as within subjects, for the go and no go, because they all did that, go and no go. The emotion, they all did the happy and fearful, but they were between subjects, they were independent measures for the high delayer or the low, because that was decided when they were 4 years old, so they couldn't run both of those, each participant. Okay, so pause me and have a go at summarising the procedures for experiment 1 and 2. On to the key findings. We look at the findings in terms of the reaction times outside of the scanner. So for experiment 1, they're not in the scanner, and then the accuracy outside the scanner, and then for experiment 2, it's going to be inside the scanner. Okay, there was no significant differences. Okay, for the reaction times, everyone's reaction times were pretty much the same. Okay, so that would be what you would expect as well. The accuracy outside the scanner, participants performed with a high level of accuracy for correctly responding to the go trials during both the cool and the hot tasks. So overall, people were very accurate. The low and high delayers performed with comparable accuracy on the GO trials. Okay, so pretty good on the GO trials. However, for the no-go trials, it was more variable. Low delayers committed more false alarms than the high delayers. So that's as predicted. Those low delayers made more mistakes when those smiling faces appeared on the hot trials. Low and high delayers performed comparably on the cool task, but the low delayers trended towards performing more poorly on the hot task than the high delayers. When you see that word trended, it does mean there wasn't a significant difference, but there was a trend in the data. The really important bit of this research is about experiment 2. That's where they're finding the significant differences in the performance. But they trended towards performing more poorly on the hot tasks than the high delayers did. Only the low delay group showed a significant decreasement in performance for the hot trials relative to the cool trials. Okay, so if you compare their performance between their gender task and their fearful face and their happy face, then the low delayers were the ones that showed that big difference in performance. Okay, but not a significant difference between the low and high delayers in any task. It was just, if you just take the low delay group, they made significantly more errors in the hot trials compared to the cool trials. Okay. Overall, therefore, the go no-go task produced differences between the delay groups only in the presence of the hot cue. So only in individuals who had a group delay and gratification at 4 years old, the low delayers found it more difficult as adults in suppressing responses to happy faces. So as predicted, those who couldn't resist at 4 years old, the low delayers, also couldn't resist responding to the happy faces. Okay. For experiment 2, similar thing for the reaction times. There was no significant difference in reaction times. They're both clicking very fast. No significant differences. The accuracy for the hot emotions, though, go no-go tasks, were uniformly high for the go trials across everybody. Okay. More variable performance on the no-go trials, 12.4% false alarm rate. So false alarm rate clicking when they shouldn't have done. Okay. As in experiment 1, low delayers committed more false alarms than the high delayers. Okay, so similar there. For the brain scans, importantly, the no-go and go trials did identify specific regions of the brain engaged at different times. Okay, so the right inferior frontal gyrus was involved with withholding a response. So this bit would become active when you were doing the no-go trials. Okay, the cognitive ability to stop yourself. Compared with the high delayers, the low delayers had a diminished recruitment of the inferior frontal gyrus. In other words, lower activity, diminished recruitment of blood flow. Okay, lower activity on the no-go relative to the go trials. The ventral striatum demonstrated significant differences as well. So the reward-related group showed a three-way interaction, group, trial, and emotion. And what that means is elevated activity in the happy no-go trials for the low delayers. So although the low delayers were having lower activity than the high delayers in the frontal lobe, in the ventral striatum, they were having much higher activity for those no-go trials. The reward, the impulses were kicking in. And so what does that show? The results showed that the prefrontal cortex differentiated between the go and the go trials to a greater extent in the high delayers. The ventral striatum showed exaggerated recruitment in the low delayers. So as I've just mentioned, if you're the Yoda type, you find in those no-go trials that your frontal lobe is much more active compared to the low delayers. And if you're a low delayer, like the Cookie Monster, you have got much higher levels in there compared to the high delayers. Okay, in the ventral striatum. Okay, so pause and summarize those results. And just join me in the final video for the conclusions.